Welcome to People in Innovation, the CPI podcast. From groundbreaking discoveries to the challenges behind the breakthroughs, we shine a light on the people driving innovation that are shaping our future. I'm Steve Bagshaw. I'm the chair of the High Value Manufacturing Catapult and your host for this series. In our previous episodes, we've explored what engineering biology is and how we scale it from lab launch to world products. Today, we turn to one of the most immediate and powerful applications of engineering biology, health and medicine. Engineering biology is rewriting the code of life, from mRNA vaccines to personalized cancer therapies, viral vectors for gene delivery, and sophisticated biologics that were unimaginable just a decade ago. These technologies are transforming the way we prevent disease, treat illnesses, and manufacture life-saving medicines at scale. And to explore this, I'm joined by two fantastic guests. Philip Probert, who's Director of Biologics and the RNA Centre of Excellence at CPI, who leads teams developing the processes and platforms that turn cutting-edge science into manufacturable therapies, and Antony Hitchcock, an independent consultant with decades of experience across the biologics and advanced therapies sector, including senior roles at Charles River and Cobra Biologics. Before we get started, I'd love to invite you both to introduce yourselves and share a little about how you came to work at the intersection of engineering biology and advanced therapeutics. Philip, would you start for us?
Brilliant. Thank you very much, Steve, for the kind introduction. So as alluded to, I have the privilege to be responsible for CPI's Biologics and RNA Centre of Excellence. And in principle, what we are doing is we solve problems that customers and collaborators who are developing novel products in order to try and get those products to the market. And we do that across a whole breadth of modalities, all empowered effectively and enabled through engineering biology. In terms of how I arrived at my current position, so I came through a scientific background, through PhD in, of all things, toxicology, for working, developing skills and expertise, and ultimately joining CPI within an upstream of cell culture capacity, based on my expertise in cell and molecular biology. And through my time in CPI and the opportunities and challenges afforded by the new modalities, I had the opportunity to establish new capabilities, support customers with development of new technologies, to my role I have now, where I have the opportunity to oversee the activities we have and see the impact we're able to have from CPI on behalf of our customers' collaborative projects that we work across. So I'm very pleased to be here today.
Thanks, Philip. Tony, how about you? How did you end up here?
How did I end up here? I guess it's a 40-year journey. I started my career at the UK blood processing authorities, working in the process development group there, looking at how we could purify blood proteins from waste blood fractions and also optimizing processes. So that was very much natural products, how we could process materials from waste. I then moved to what was then still ICI Pharmaceuticals and now AstraZeneca. And at that time, ICI were working, I guess, on very much cutting edge biotechnology, where they were actually looking at taking native molecules and modifying them through amino acid changes to look at stability, improve performance, and how we could use those. And also, we actually worked, I guess, what was highly pioneering work on things like antibody drug conjugates, very early ones. People think they're new, but they actually go back over 30 years, and it's taken us that long to get them working. After 6 years there, I got the opportunity to move to what was a very small startup, which had been set up by formerly our head of biotechnology, Roger Craig, a company called therapies. And we were looking at what was then very early gene therapy, looking at using direct treatment using plasmid DNA and some conjugations. And that was very much new and novel. And I think at the time, the reason myself and other colleagues moved, what was we started thinking, why do you need protein purification when you can do that as a single treatment and it's a curative therapy? And I think at the time as well, we were making novel de novo proteins out of very much platform processes, so developing processes for new products just to get into the clinic took several years, and it was hugely expensive and only really accessible to large pharmaceutical companies. So this was a very novel opportunity. But as we moved into that, we started to realize that we couldn't actually achieve the delivery that we wanted. We could do ex vivo studies, but in vivo didn't work. We started then looking at viral vectors as a means of delivery. And I think we can come back through that theme that it's not just coming up with a therapy, but how do you deliver that to the patient? And I think that's a really critical element of the product and processes. And again, we saw failures in those early gene therapy companies. And as a get out, we started doing contract manufacturing services, and that was called Cobra, which actually got nothing to do with snakes, but was taken from the Clinical Oncology Birmingham Research Associates as we bought a viral vector company. And so we started looking at having the skills to make plasmid and viral vectors, how we could do that as customer services. So my involvement was very much about how do we develop the manufacturing processes for plasmid DNA to get that to clinical grade, and also working on viral vector products, things like adenovirus. And then more recently, we started realizing that plasmid, rather than it being a direct therapy, the early uses were actually things like HIV vaccines, TB vaccines, and other therapies. We could actually use this as a critical starting material for making things like AAV and lentivirus. And that's kind of tells us where this is going. So we now have, one of the things I was very much involved in is developing platform processes for making plasmid, how we could optimize those processes and scale them up. And the same looking at how we develop processes for making vectors. So I guess that gives us an introduction in terms of both the move to curative therapies, but also the move from having completely de novo processes for each new product to actually establishing platforms where we can have a platform where we can make multiple therapies and rapidly translate from early stage development into clinic and then hopefully to commercial production. So that's been my journey. And I now, for the last 3 years, worked as an independent consultant, working for companies in the UK, but also in India, the US, and quite a broad customer base.
Tony, that's brilliant. And you've given us a real sense of the history, if you like, of the step-by-step, how some of these products are developed. You didn't use the word engineering biology once in that. I wonder, Philip, if you could perhaps use the word engineering biology to describe how this is, how is engineering biology, to take Tony's story, how does it fit in and how does it all fit together?
So perhaps, as you know, to consider the first biologic products, if we think how it's so impactful and why engineering biology it's been so important in terms of modern medicines. If we think about the insulin as an example, it's the first kind of biologic medicine. So back in the 20s, the Nobel Prize awarded to Banty McLeod in Toronto, which I had the privilege of seeing in Canada a couple of years ago. I've got a very flashy picture of me standing next to it. You know, they were effectively identified insulin as being a treatment for diabetes, but at the time was being generated from pancreas or from animals, purified and then used to treat patients, which, you know, saved lives from patients on wards. What engineering biology enables us to do is, through our understanding that we've built in that time, in the last 100 years, effectively, in terms of understanding the role of DNA, which makes mRNA, which makes proteins, and the code of life, as we call it, we can then utilize that and leverage that to our advantage to make these things in a more cost-effective, higher quality, and a more ethical manner. So if you think about something like insulin, so it's the first approved biologic in the early 80s, we know the sequence of insulin, and we're able to take that sequence, we're able to synthesize that chemically, insert that into a cell, for example, an E. coli, a bacteria cell, and then get that cell to make lots of insulin. And we can then purify that product in order to then create a recombinant form, so recombinant DNA technology, which facilitates and enables the first real biologic products. So while that's really effective, is that we are all people, but we're alive. We all have molecules in our bodies. By understanding those and then leveraging that to then create more of it, or in a more targeted manner, we have the ability to have the incredible impact on medicine that we've seen. So from insulin, and then through all the products that we have now, you can see the impact, and that ultimately is all enabled through how we've engineered biology, particularly of other cells and systems, to create particular products that we've used to treat ourselves, and have the impact we've seen on modern medicine.
No, that's brilliant. And Tony, if you build on what Philip just said there, in terms of the career, the story you just told, why in the last 10 years has mRNA and viral vector technology has been such a leap forward? Because in a sense, all of what you've described has been leaps, hasn't it? If you think of where we were 40 years ago, the huge leaps that we've taken in our understanding. But why the last 10 years have we got excited about mRNA and viral vectors?
I guess the key thing is actually understanding, one, they're not necessarily new. So as I was saying, these have been aware. I think the issue is about understanding the biology, about how you make vectors better, how we can target systems, and also the opportunities with those products. So it's actually, how do we manufacture them to the right standards? How do we build them at the right scales? A lot of it's about overcoming that knowledge of biology, of how we can actually make these products at a point where we can actually consistently put them into patients safely. So a lot of it's about the underpinning. Philip talked about, the knowledge of this, but we also need to understand the manufacturing systems. How does a cell make a virus? I think when we started 10, 20 years ago, people saw us as a black box. We're now taking that apart and understanding how we can actually control and, whether we can modify cells, we look at producer cells, we can engineer cells as factories to make these kind of viral vectors. And the same with as Philip can talk a lot more than I can about the RNA products, is how we not just make them at small scale in labs, but we can actually make them at the scale and quality we need to be able to use these as medicines. So it's the manufacturing, but also looking at, how can we best use these as therapeutics rather than just having the technology to make them? What is the best applications for this? So I think it's that overall underpinning knowledge that's really opened up the opportunities for these therapies.
Brilliant. That's really understandable. And Philip, I'm guessing if you ask most people on the street their understanding of how medicine's made a difference in the last 5 years, then someone's going to mention COVID-19 at some point. And I remember the summer of 2020, people asking us, could we really get a vaccine created in 8 months? How did mRNA and engineering biology help for us to get from where we were in the spring of 2020 to December 2020, having approved vaccines?
Yeah, absolutely. I mean, it was certainly, I think, for many of us, quite a very eventful period of time for a multitude of reasons. So, we, as Tony's alluded to, RNA technology per se isn't a new technology. And why mRNA technology is really powerful, because you can generate a lot of material very quickly, but that's enabled by the fact we have a great understanding of all the steps it takes to manufacture these products. So we think about how we manufacture mRNA. We use enzymes to manufacture mRNA, and those enzymes are all themselves coming from cells that we've all engineered to make these particular enzymes and purified. Same with some of the other components. So what engineering biology enables us to do is that through the understanding of the technology, able to take, say, proteins we've engineered, purified, we're able to take the sequence of, say, with coronavirus, there was quickly scientists were able to understand what might be an effective sequence to vaccinate against, to build the antigen on. So again, we took that understanding, so we could then create the DNA template that we use to make the mRNA, and be able to use the expertise, but also the expertise built in the last 40 years or so around making biological medicines, to actually very quickly, and was incredibly quickly given how long it typically would take, to take this new technology and to overcome some of these scaling challenges. But I think, again, benefit of RNA, and because of how we manufacture, it's an easier scaling approach, and it's a quicker scalable approach than some other technologies. And fortunately, in this case, as we saw in the case of the Pfizer and Moderna vaccines, the vaccines were really efficacious, and through to the ease of manufacturing, we're able to then quickly scale up and manufacture these medicines. And again, we're able to build on that now in terms of how we manufacture.
Sorry, Philip, that it was life-changing, wasn't it? Or life-saving, I should say, for many people, what was done there. Tony, looking forward, what do you think are the most transformative uses of mRNA vaccines going forward at the moment, and in terms of the ways in which we're using both mRNA and viral vectors? What do you see as the most transformative?
Certainly in the case of things like the RNA vectors and also some other vectors, I think it's our ability to do things like modify immune systems. We're looking at, cancer vaccines, how we can actually use these kind of therapies, not just for disease, infectious disease, but one of the origins of people like BioNTech and Moderna was very much, and CureVac was very much developing, can we use these kind of vaccine systems to mobilize the systems to support immunotherapy and oncology? And I think that will be one of the really great opportunities. And again, Philip talks about, the broader knowledge. We can now get to a point where we can personalize those therapies. So people are looking at, how can I make a personalized cancer therapy? So can I sequence your tumor, identify potential antigens, rapidly make a vaccine using these kind of platform RNA technologies, and then use that in combination with the other immunotherapies? And it's something I think the UK government's been extremely good at. We've got long-term trials going on with BioNTech on this. And so those can be potentially transformative in terms of both the outcomes and the experience patients go through with these kind of treatments. So I think it's those types of vehicles that we're looking at, how we can modify cells, how we can increase, cellular performance of things like the immune system to try and address these kind of diseases. I think those to me are the really kind of key areas, and people are also looking at things like autoimmune diseases. Again, how can we modify immune systems? So I think those are kind of the key areas I see these kind of developments over the last 5-10 years.
What do you think the barriers are that will stop us from getting there?
I think for anything about personalized products, and particularly talking about mass personalization, I think there's cost, the ability to manufacture these products quickly in a cost-effective manner. Ostensibly the manufacturing itself is not an onerous process, but because of the requirements for it to be safe and it's to be proved that what you've made is what you said you're going to make, there's a lot of quality checks, which if you're making a single batch maybe once a year, a few times a year, and particularly where you've got less time pressure because you've got less critically ill patients, you can facilitate that. But when you've got patients who are critically ill and you need to make multiple products, even a single day and a week, you need the ability for that to be quick, so you can get the medicines to the patient quickly. But you also need it to be cost effective, so that people, in a very dry sense, health agencies can afford to pay for these medicines. And there are opportunities, and as Tony said, I think the UK is making real strides in this space. But it's how we try to balance the need for speed and critically the need for safety.
Tony, anything to add to that in terms of what the challenges are ahead if you look forward 5 years in?
I think it's actually almost a change in mindset as well with these types of therapies. Historically, we've always developed processes which are scaled up by, we just buy bigger tanks and bigger processes. This is, you're scaling out horizontally, and also even for non-personalized therapies. So we need to also look at how we can use automation, data control, knowledge. So these processes become a lot more fully automated. We have online data collection, online quality systems, and probably move away from these kind of historic systems where we've always had to make a batch, then spend 3 or 4 weeks, months testing, analyzing the product before we trusted that the process we've already validated works. One of the real challenges is, can we develop processes which are so well controlled that we don't have to go through those kind of expensive and time-consuming analytics of testing a product? Can we develop processes that we know the outcome we can control and understand? So I think it's a great opportunity, but I think both from a manufacturing and also from a regulatory perspective, we probably need a complete change in mindset to a very different operation than we have before. And I think the mRNA platform does offer that opportunity of how we can, make those massive step changes in how we go about manufacturing products.
Do you think, Philip, from your vantage point, do you think the challenges you've both laid out there of the difficulties of personalized medicine, do you think they're overcomable?
Absolutely. I think when you look at, some to Tony's point over, the challenge of scaling out and the, what is a manufacturing scale, the technology is coming through now that will facilitate that. I think when you look at what you may need to do in terms of the regulatory component, the quality component, again, I think there are pragmatic solutions that will facilitate both the cost and safety, and actually, which have been enabled by advances in technology in the meantime. For example, how we use robotics and automation, how we've managed to scale down things like DNA synthesis systems. So I think if we look towards the factories of the future, which again is a novel term per se, but you can envisage how some of these medicines can be made, and that's not only facilitated by the progress within manufacturing, but also within diagnostics and other things, which are all part of how we manufacture personalized products. But I think it requires multidisciplinary teams. It requires that common sense of purpose, and it requires obviously buy-in from regulators within nation states to ensure that you can get the approval to then enable these medicines to be available to the public where they're most needed.
And when we talk about medicines, just thinking about what you just said there, Tony, one of the things I really liked about when you did your introduction, you talked about curative disease, curative medicines, and the difference, if you like, from where you'd started your career to where we were going. When we talk about personalized medicines, what's the opportunity for curative medicine here?
Well, clearly, in those kinds of areas, we're looking at, having a much deeper understanding of your nature of your disease. We've already got curative treatments in potentially things like the CAR T therapies, which may, be using your personal cells. But we're also looking at not necessarily personalized, but curative treatments in terms of direct gene therapy when we're looking at gene replacement. So, these have, rather than people being locked into long-term treatments and chronic, injections, people like hemophiliacs having to continually inject factor VIII and carry those around, we can transform people's lives. So I think it's both personalized, but it's also transformative. CAR T therapies seem to be curative. People are not getting returns of cancers. They're having a profound effect on, they're clearly very, very poorly people as the last line of treatment. So, and then again, they are based on user processing and are using a patient's own T cells to develop these therapies. So they are there. It's just how we get those to more and more patients, and looking at the broader range of diseases we can actually treat with these kind of technologies.
Yeah, that's interesting, because I remember reading about how gene therapies were targeting rare and ultra rare diseases. So do you think there's a chance that, what Tony's just been talking about there is very much about more mass availability of these therapies? What do you think is important for us to get right for these to be more available?
Do you mean in the context of for ultra rare diseases, or for
no more to what Tony's really talking about is use CAR T therapies. They tend, we're starting to look more at cancer and things that we might all get.
So I think taking CAR T is an interesting example, and it's within a CAR T therapy, you're taking cells out of the patient, you're then doing the genetic editing, and then you're doing safety testing, and then you're reintroducing those cells back in. And again, as Tony's alluded to, really efficacious, really novel. And there's other similar types of stem cell therapies within the kind of cell therapy space. I think what we're starting to see interest in, and this is enabled by other technologies such as CRISPR-Cas9 type things, so tools which enable you, engineering biology tools which enable you to make specific edits within cells, is the ability to actually avoid the distress and additional work required to pull those cells out of the patients and actually go straight to those cells in vivo. And because of, something like CRISPR is you can make purely synthetically, so again, you can move more quickly. And if you with the exquisite sensitivity from some of these tools, enable you to then go directly to the patient and make these edits directly in vivo. And again, if you think about the power of that, you might turn up with you might have a particular genetic disease, you might have a really terminal cancer. You turn up, you get an injection, and actually you're cured. And it's imagine the quality of life to the quality of patient's incredible. It can be delivered relatively flexibly, and fundamentally it should be more cost effective. What a thing to be to be enabled by the technology.
Yeah. And in terms of, Tony, you talked about platforms again in your introduction. What are the platforms that you look at and you think are the most promising as we go forward?
Clearly, in terms of the platforms, we've already got platforms for making plasmid. We can make that bacterial and now synthetic approaches. There's much more novel systems coming out of making plasmid. Clearly, one of the things about the RNA when we talked about getting to COVID, one of the things that allowed that acceleration was that was already an established platform process. We didn't have to go back and reinvent it. And the same is applying to these viral vectors, whether you make adenovirus, you make lenti or AAV. They're increasingly standardized platform processes. And I think, as I could probably mention, that one of the key things about that is also being able to rapidly get products into the clinic and actually understand success and failure. One of the key things with all these kind of therapies is actually how can we start generating data and understanding the knowledge of them, and how we can also reduce the cost of getting those products into the patient, and that, rather than being a 5-6 year journey, this can be a 2 or 3 or less time. So we have very little time optimizing processes, and we can allow these therapies to get into patients. And I think that's the key thing about the platforms, is both having them and then how we best exploit them in terms of getting products to patients.
Yeah, and that sounds really promising. I want to move us on slightly and talk about something that I know, Philip, is close to your heart. You've got an RNA Biofoundry. I wonder if you could start at the beginning and tell us what an RNA Biofoundry is.
Absolutely. So Biofoundry is a big term in terms of what it means in practice. So when we look at how we, for example, to date have manufactured RNA, there's kind of a range of different approaches. So lots of different organizations use lots of different bespoke approaches. But the challenge is, often it can be expensive to make material, and there'll be distinctions in terms of quality arising from different types of process. And ultimately, it also can take quite a long time because of the time required to kind of develop and move products onto these platforms. The purpose of the Biofoundry is to look at how we make RNA currently and to do that more quickly, to do it more cheaply, whilst maintaining or enhancing product quality. So what the Biofoundry is effectively is a series of cutting-edge technology which enable us to go from a digital sequence, so a sequence for a particular gene or antigen, and then make the DNA, make the mRNA, and encapsulate that all through a standardized platform to effectively enable us to make material for organizations, be it academics, be it small companies or larger pharma, much more quickly, to great quality at much greater pace, which will, catalyze the market to enable more of these products to get to people where obviously they're most critically needed.
But how will you use having an RNA Biofoundry? How will you use that to sort of push the UK's position as a place to develop RNA medicines?
So what it will enable us to do in the UK is, all companies and organizations are looking for suppliers, are looking for where to base their operations. Within some of these technologies, it's important that before you can get to the clinical stage, there's all the early stage development as well. By working with us, we can support that early stage preclinical work, whilst also then supporting the clinical studies. And with support of the UK regulator, who again, we're seeing great engagement from in terms of trying to make the UK the place to do trials, and even announcements I think this week about further innovations, we can create the environment from both a manufacturing perspective, but also the right regulatory framework to make the UK the right place for companies to establish themselves. Whilst equally we'll also provide material overseas to support overseas innovation.
Right. And if I'm an SME or an academic in this country, how do I actually get to access what you've got?
So what you get in contact. So the wider Biofoundry will, in terms of the form of Biofoundry, will ultimately have a brokerage service. Our companies will get in contact with when the full service is live, to then facilitate access to material. Outside of, for more bespoke programs, again, we obviously within CPI have conventional offerings. Again, we're also able to support to provide in the meantime whilst we establish the Biofoundry program.
Great. Now, Tony, where you sit, how important is it for the UK to have assets like a Biofoundry?
I think, as Philip mentioned, I think the one thing is to try and attract companies into this country. We probably haven't done as well as we ought to on larger scale biologics, but I think these novel therapies create us a new opportunity. And I think a lot of it is about, it's not necessarily about big capital investment, big facilities. It's about the skills and the knowledge base around it. I think Philip mentioned about, it's the breadth of skills as well. It's not just a skill in a certain area. It's about development. It's about understanding the manufacturing, the quality, the regulatory systems around that, and building that really broad skill base. Because I think these products we're talking about are not going to be made on multi-thousand litre scales. It's about flexible scaling out, having the right management skills, the right operational flexibility, and it's ultimately about the people business. And I think if we can show we have the right people, and also the right environment of government support, skill base coming out of academic and universities, then we have a great opportunity to develop companies in this space.
And when you both think about the world that you've worked in and you're working in, what is the biggest hurdle a company faces in getting from a really good idea academically to making a medicine that starts to either cure people or to allow people therapeutic relief? What both of you have a chance. Philip, you go first, and then Tony.
I'm going to be cheeky because I'm going to get two things.
Yeah.
The first thing I think, and this, as someone sitting in CPI as a centre designed to do this, it's having access or cost-effective access to the right skills and expertise to then take a great idea and scale it, because the skills and expertise associated with discovering a product and understanding the mechanisms of action of that are very different from how we actually manufacture that in practice. So I think there's that piece, and someone at CPI established to support companies with that. We see every day. But I think the second point is the access to funding to do that. So it's expensive and it's really risky. Most products going to trial ultimately will fail. Now, if you get a successful product, they can be worth billions of dollars per year in terms of worldwide sales. So there's a high impact you can get it to market. There's a high risk to get it there. But I think by having the funding and having the access to the skills and kit, frankly, you can enable that. But you need both those things to exist.
Great. Tony, how about you? What would you say?
I think that I'd kind of build on that. I think where people go wrong is that they feel they can make something in the lab. So why is the big challenge of trying to make that at a large scale in clinic? And people think it's just about having bigger tanks. But I think Philip alluded to it before as well, as a clinical process and manufacturing products for the clinic. It's about quality, consistency, and safety. And that often involves going about and stripping a process back to the very basics and trying to understand each step, controlling, developing that so we can have consistent outcomes. Your former colleague, Mark Carver, used to talk about understanding the underpinning biology. And that one of the issues, I think, again, about viral vectors was we didn't understand the vector processes. We just thought we throw a load of plasmid into a shape flask, with some cells in a transfection, and lo and behold, we get virus out. But there was a lot of problems about the consistency, the variability, the potency of those products. And it's that kind of changing the mindset so people understand and appreciate, as Philip mentioned, it's a different skill set. We need both the development, but we need the skills, knowledge, and experience within the manufacturing environments to understand how we go about that. And I think if you try and shortcut that and try not to, it's painful and sometimes expensive and time consuming, but if you don't, you get punished later on. And I think that recognizing the breadth of skills to get a product of the right quality, the right consistency into patients. Because I think one of the problems we had in the viral vectors field, for instance, was there's a lot of variability in the quality of products, both in the purity and safety of it, but also the potency. One batch was good, one batch was bad. And it's because we didn't have that knowledge of the virus production processes and the analytics to support that we could characterize and understand what we're getting out of it. So it's building all that together and recognizing the need for that, I think, is often the big hurdle when people kind of, I don't want to pay this, I don't want to take that time. But we've all seen it. Everybody rushes through, goes through it, and then it gets expensive when it goes wrong.
So you've given, I'm sensing you've almost given us a reverse engineering definition of engineering biology there, of why it's so important to understand the constituent steps of a process and be able to analyze them. Philip, there's an awful lot of data in all of this. How does data and handling and digital steps forward, where's the crossover here between some of the digital advances we've got and our engineering biology knowledge?
I mean, certainly from my perspective, you've got the innovations around, so say, some of these novel products looking at, there's the ability, particularly personalized products, you need the ability to design the personalized sequence against a particular cancer that a patient might have. And that ultimately facilitates things like machine learning type algorithms, where building the data and understanding mechanisms of action, companies are then able to apply that to then engineer essentially a product. But we also apply, and there's a lot of great deal of interest due to the benefits it can afford in terms of how we better control processes. So, manufacturing any product is a big exercise. It's expensive. There's a lot of risk, and biology is exciting, but it's also quite difficult to control. So by using data, we can collect the data, we can build models of data and use that to enhance how we control processes, so we can better predict things and stop them going wrong before, the process runs away with itself. And the challenge with all these things is, there's data, there's so much data, huge quantities of data. It's how you use that data in a way that's genuinely value adding as opposed to a distraction. And I think it's only now people are getting more of a handle on it. 10, 15 years ago, everyone was excited by, you had these data lakes, data reservoirs, there was all these terms, but data wasn't really doing very much. But now we're getting better and enabled through, things like AI tools are helping how we conceptualize and utilize that data to actually do something useful. The so what is, why is this data useful? It's useful because it's doing these things for us. So really important, still more to do, but a real area of innovation currently.
Tony, have you got a view there?
I think I totally agree with that. I think I've been through a period as well when you talk about the barriers of developing this, people somewhat don't see the value, especially when you're doing process development. You're collecting now, for the mention, we have tools, things have moved on. We can run 24 bioreactors simultaneously. We can collect huge amounts of data, and people are often dismissive of it. I've worked with customers and they see no value in it, but actually there's a huge amount of knowledge base in there, and we can use that to control processes, accelerate development, and really understand how the biologic systems are working. And I think that's when talking about engineering biology, how can we make cells make better products? How do we understand that? We have the tools to generate a huge amount of data, both within the process and from analytics. And I guess it's for developers especially to recognize the value of that data and understand how we can best use it and work with manufacturers. Do that. It's, people tend to take shortcuts, but we now have systems that generate so much data. We have, the opportunity to really start interrogating and understanding our processes a lot better and control them and scale them up and deliver, much faster to the clinic and to the commercialization.
So how competitive is the UK at this?
I think we're getting there, might be a way of putting it. I think we've all been in meetings where this was kind of first discussed, but we're now seeing companies taking this a lot more serious. It is a significant investment in terms of going to digitalized processes, digitalized batch records, digitalized systems, and it's actually having the skill base across the companies. Biologists are not natural IT guys. They don't necessarily understand that. They get surrounded by people using a whole load of terminology they don't understand and they don't follow it. So it's difficult to make business cases around that. But I know companies are doing that. They're getting support from the government to do that. But I think it is really important that we start recognizing the value of that and show the value of that within companies and to customers.
This discussion is great, and especially the way the UK is heading. I wonder how we take the public with us in this. So medicine hasn't really been scrutinized in the way some of the other uses of engineering biology have. But how do we continue to build trust with the public in terms of the way in which engineered medicines are developed and made accessible? Do you have any views on that, Philip?
I think it was interesting over the course of COVID, where suddenly there was a lot of interest in the sector, certainly more so than ever seen previously. A lot of questions about these products, what they were, how they were being made, and there was it was a great opportunity. There was a lot of education, but also because there were a lot of misconceptions about what some of these products were, and some of the words we use. Engineering biology is a big phrase. It sounds like it's a bit wary. Like, well, what does that mean? What are you engineering me? I think it's important. A lot of these things, I think, it's education. It's the impact. I think, we've had this conversation. A lot of stuff we're doing here isn't new, but we're seeing an acceleration of some of the stuff we're able to do. So I think it's reiterating that some of these things are not new things per se. A lot of us have seen the impact. A lot of us have had family members who've been unwell, who have had some of these type antibody products that have also been have treated them and have cured them of a cancer. So how we educate, how we reiterate, the fact that we're building what we've already been doing for decades now, but also what it will enable us to do in the future, the impact it will have in real world, like what it means for you, for me, by manufacturing these products.
I think that the way we explain it, the way we're prepared to talk about it, seems really important. Tony, when you were talking earlier about cancer vaccines, I was struck by the fact that if you're listening to somebody talking about personalized cancer vaccines, they sound almost a bit like science fiction. When someone stops you in the street and asks you, what is a personalized cancer vaccine? How do you explain it?
I think you have to explain it step by step and factually. And I think, that is the point of actually not having misinformation, having accurate information that we can present critically to patients who are likely to receive these therapies. So I think, how clinical centres talk and handle it is very important. I think one of the things about personalisation as well is the ethics about data handling, that you're going to sequence someone's genome or tumour's genome. And there's clearly been issues about that, of how we handle issues about confidentiality. So I think I can see issues arising around that. Is that done by the healthcare provider? Is it done by the companies developing the vaccines, for instance? Who owns that data? I think things like that are critical. I think the other things that come in there as well is, we demonstrate patient outcomes. We're clearly looking at CAR T therapies where we modify people's cells, and that gets in the news. And there is some explanations of it. Some of the news or corporations put quite good explanations of that. So they do try and do that. But clearly the key thing is about, what's the patient outcomes from gene therapy, for instance. You see children with SMA starting to live normal lives. I think that's the biggest thing. If we can show, these are positive clinical outcomes, these transformative medicines. The public do have a relatively low understanding of that, and I think it's difficult to go through the science at times.
How important is regulation? Sorry to interrupt. So as the public does what it does, what's the role of the regulator in this? Then do you think?
I think for me, it's very important. I think there's two elements of that. One is that we invest and support the regulators, because the one thing we've talked about, this is a very rapidly changing field. So the regulators need to have the skills and the people within their organizations that understand these products, the risks associated with them, and how we can best ensure safety to the patients, but also how they can work with producers and developers to set appropriate regulations to allow these therapies to come to the market. Because it's one thing talking about the science, but if we can't get them to patients who desperately need them, then we're going nowhere. So I think we really need to invest and develop the regulatory frameworks around that so it works for everybody. It's critical that we have the environment where we can develop these processes and products, and that those regulations, they work hand in hand with manufacturers to evolve and develop those therapies. But I think with so many different therapies coming through and so many different technologies, it's a real challenge for the regulators to retain the required skill base and knowledge base. And so rather than cutting back on that, I think we really need to invest and develop the skill bases within the regulators. So I think that's absolutely critical, and it's critical to public trust as well.
Brilliant. Thanks. Philip, what's the role CPI can play here with the regulator?
I think CPI's, our role is to support companies with, novel, challenging products and processes. So we have the opportunity to be, we get to see a lot of things that are coming through. And we saw, for example, of when working with RNA, that we had the opportunity to actually support the regulator in terms of understanding the technology. I think similarly with new things coming through how we understand particular challenges that we see and our understanding of what is going to come through, to be able to articulate, explain, and help to kind of demystify, and perhaps hopefully help to influence and support the regulator in terms of how we support these companies with their products. And we can take that learning back to the companies we work with to support them in terms of the things they need to be aware of, that they're going to be asked about, which they need to do in order to get their products into patients.
Yeah. Thanks. Okay, a question for both of you. One of the ways you can tell whether the public are supporting you or is almost a bit like the supply of skills into the industry. Are people interested enough in this whole area? What do you think we need to do to get more people choosing this whole approach as a career? Tony, I'll let you go with your long look back over your career. What do you say to people who ask you, should they do work in this area, and what more should we be doing?
Clearly, I think it does have a lot of career opportunities to develop people's breadth of skills and opportunities throughout the industry. I think it is a great industry to work in, and it's very rewarding in terms of we engaged in the COVID projects. That was very rewarding for people, as well as other therapies that I've worked on and seen how they've transformed people's lives. I think it's a long-term thing of where we need to engage at all levels, at schools, at academic centres. We're relatively good at that, but I think we could do better, especially within schools and understanding knowledge, and also trying to understand what are the appropriate skills people need, and we need to not trend that. I think it's more, we need people with good science skills. We've just talked about data handling, data management, people who can do all that kind of AI stuff. And actually, those are the kind of people we need to attract, and again, show that to people. So we probably need to be broader in the way we look at the skills that we need to come into the industry, rather than just looking, I need a chemistry or a biology degree. We need to be a bit broader and actually show the opportunities to companies. I think, we have some good apprenticeship schemes. We need to develop those and expand those. And again, the ability to reskill and retrain people into the industry, again, bringing people into that. So it's a long-term thing. I think it's also one of understanding as well, not governmental, but also regional. We've seen a lot of success, for instance, at Rosslyn Cells in Scotland, you both know well. But I know, Peter Coleman put an article out in the Guardian last week, of how much Scottish government had actually supported them in terms of doing their skill base. And I think things like that, people within those kind of areas, it's not necessarily their key area, but actually understanding the opportunities within these sectors to actually try and expand and develop, new modern industries in their areas to try and, give opportunities for graduates and others within their kind of regional. So I think it's a regional as well as a national thing.
Yeah, that's great. Philip, what about from your perspective?
I think, what people want to have in their careers, I think obviously beyond financial and the remunerations, I think they want to know they're having an impact and, that what they're doing is important. I think one of the challenges around that, I think that's some of the work that we do. I think, COVID's helped, and, things like Ozempic, Manjaro, suddenly everyone, people really, where things have impact, everyone's talking about them. But for a lot of stuff, actually, has been very impactful even for the last, several decades. People don't necessarily know what these things are. So they need to, people need to understand the impact that these things can have. I think they need to, and then that drives interest and actually, that's something I want to be involved in. And then it's understanding how to get involved in that. And I think there's been a lot of interest in the last few years. It's how we try to develop more specific and bespoke training programs to enable people to step into these types of careers. So, so Tony's mentioned, we found apprenticeships to be incredibly impactful because you've got on-the-job training and it's specific, to what people want to do. But it's also how we ensure that these other types of, like biology, chemistry, data courses, they have the right transferable and translatable skills and people understand how those things will enable them to step into these types of careers. Because there isn't a biologics degree, but there is a law degree. So how do we try to make it easy for people to step into these jobs, which will help, again, supercharge the UK because we'll have the people, because people drive action. So with the right people, you can have an impact. Without the people, you've got nothing. So we have to focus on not just what we have now, but how we make it easy for people to do what we perceive to be the right thing so we can make these, incredible medicines.
Yeah, no, I really like that. So you get people excited about it. One of the things that can get people excited is, as you've said, I think what you're doing with the products that we're making. But what does the next 10 years look like? So if the pair of you were now sort of engaged in a bit of future gazing, what do you think 2036 looks like in terms of where we'll be and what might the medicine and health world look like back then? Tony, do you want to go first?
I guess the one thing we talked about, data and knowledge and big knowledge about disease understanding. And I think that's probably the one thing that's going to change is we have, and I think the UK has been working with that. And although there's some controversy about it, of actually accessing the huge amount of knowledge and data we have about disease processes, potential therapeutics that become increasingly personalized. We have that level of knowledge, we can stratify disease as well. So we have more medicines, but better outcomes. Clearly, we need to learn how quickly we can use that. Things like AI are likely to have a profound effect on drug design, drug development. We're going to see much more rapid development of that. And also, I think, clearly there's going to be development in oncology. Cancer is clearly going to be a big area, but also these degenerative diseases, Alzheimer's and various other ones like that, where we're gradually getting an understanding of those kind of diseases. And I think it's going to be in those kind of areas we're going to see that. And also things like the autoimmune diseases, which affect a very large number of patients, and how we can, again, we talked about, understanding of the biology about things like immune, how we can modify the immune systems. So I see it all coming together. We have, we can genetically sequence someone in about an hour now, apparently. We have a huge amount of data and knowledge. It's going to be how we best use that to improve patient outcomes, both in terms of drug design, but also the therapies, how we diagnose disease, right through to the development and delivery of those treatments to patients.
That sounds optimistic. Philip, are you as optimistic as Tony is?
I am optimistic. I think we have to, 10 years in the world of medicines and manufacturing is not a, there's a lot to happen that will happen beyond that. But I think equally it's about the path. It's about setting the path and where we are on that path. I think when I think about us in terms of, how we operate, I think it's trying to move from, medicines that we manufacture, that we apply to patients, to patient-led medicines. So how do we, by more truly understanding the patient, and again, it's not just about manufacturing, it's about how we diagnose patients, which Tony's alluded to. Like, diagnostics are flying through, like incredible innovation. So more feasible to stratify the patient population by developing products which are both more potent, but also have fewer side effects. And that's through a whole wealth of tools available to us. But one of the areas I'm really excited about is when we talk about, these MPICs, your GLP-1s, that's effectively, it's a hybrid of biology and chemistry. It's got natural things, it's got unnatural amino acids factored in. How we hybridize biology and chemistry and bring together small and large molecules to genuinely drive innovation within medicines with exquisite sensitivity, but also incredible potency, which also drives cost because it's too expensive to no access. So it's got to be cost effective to access. And on top of that, we can also look at how we intensify, so we reduce the carbon footprint of how we manufacture. So I think, there's a lot of things happening in the space at the moment. I think there's a journey to get there when, 2035, we'd like to be on that journey. We're not going to be at the end of that journey yet, but I think there's incredible activity and innovation to take place and I think that people will feel it. And that's the important thing, is we can talk about, but people will feel the difference in their lives in the same way that, people feel the difference that a GLP-1 agonist, Ozempic have. You can see it. So how do we drive innovation that people understand and feel? Because again, to that previous point, we also want to have the skills coming through that will facilitate that future growth.
Yeah. And it's interesting that you've as we come into the close of this conversation, Ozempic and Mounjaro, we talked about today's story, aren't they? The story of the vaccine development was 4 or 5 years ago. But I think through both of those stories, you can see how the impact on the public becomes something that makes a difference to how the way people perceive what we're doing. In a way, perhaps people take for granted now that breast cancer is no longer a killer automatically because of the biologic drugs advances we've had, really, and the understanding of the way in which we can use therapeutics. So no, I think that's, both of you have been very optimistic there. I like that a lot. What do you think? So can you keep your optimism going by talking about how the UK builds a resilient and competitive ecosystem to do this? How do we do that, Tony? What's your message?
I think we've talked a lot about skills and developing the knowledge base, because I think, those are going to be critical to that. I think it is also, government regional support and understanding the value of life sciences within people. I guess one of the challenges we need to do is also try and get investment in terms of UK-based investment. A lot of what we do seems to rely on US, Japanese, various other companies coming in and investing in the UK, and actually have the confidence of UK investors to actually develop. We have companies set up in the UK and actually see UK money investing and supporting us, rather than being just an outshoot of some company based in the US. So I think, recognizing the value of the life science is absolutely critical. So, we do start developing facilities within the UK, and that's supported by, let's say, UK investment and understanding, it's not just about early startups and venture capital, how we can actually develop companies that are long-term based within the UK. And I think, that's about mindsets as well as government policy and support.
Yeah. And ecosystems is a really interesting how much you need to invest also, not just in what you talked about, but the supply chain as well. So you've got skills, you've got supply chain, haven't you, in terms of the ways in which we create a resilience. Philip, any insights from where you're sat?
Yes, I mean, I think maybe to, build on what Tony's indicated, I think, you need the science, you need the people, you need the right kind of technology, you need the right regulatory framework so that you can manufacture, but also you can get it into patients quickly, and then hopefully get approval to actually commercialize the product. And it needs to make commercial sense. So the finances need to stack up. So what kind of level of relief, what kind of investment will be available to try and house companies in the UK. I think the UK is not a big country. everyone's relatively close together. Small countries need to be more agile. We saw this in the pandemic where, the AZ vaccine with Oxford, that was through people having the networks, knowing each other, driving an activity incredibly quickly. So how do we leverage our ability as a country? How do we focus on what we want to deliver? And how do we then do that? And we can't do everything, but by being choiceful, we can build, with the ecosystem we have, fantastic science in the UK. We can build an ecosystem that drives value for the UK PLC. We're seeing some interesting movements with MO and people, I think, aligning within that, within government. It's continued to drive it. And it's through political cycles as well.
Yeah. Well, that's a really positive way from both of you. Thank you to end this podcast, People in Innovation, a CPI podcast. We've explored across the world of therapeutics and into today. Tony started by taking us through a 40-year journey of where we were 40 years ago, and how the acceleration of the use of engineering biology has happened, and how therapeutics and the ways in which we use the mRNA and the advanced therapies, how we're using them today. We've finished there with some fantastic thoughts about the future. We've continued to talk about people at every stage. It's been people that's got us to where we are now. It continues to be people that are the important part of our community and the ecosystem to drive this use of engineering biology. We've also looked at the things that need to happen to actually make this a reality and make it more accessible in terms of the use of these kind of ways forward of using mRNA and the sort of advanced therapies. How do you use those more? And we've talked about regulation and the way in which we need to reduce cost and increase speed in terms of the way in which we change the way that these therapies are used. So it's a huge thank you to Philip Probert and to Antony Hitchcock. Thank you for joining me. Thank you to those of you who are listening and watching this podcast. To learn more about CPI's work on engineering biology and advanced therapeutics, visit uk-cpi.com, and don't forget to subscribe so you don't miss the next episode, where we'll continue exploring the innovations that shape our future. Let's innovate together.